Yes, so I'm excited to present our work on the cachexia lipidome. Before I start going into the details of this work, maybe I should present to you or introduce to you what cancer cachexia actually is. My group is studying metabolic diseases, and cachexia is one of them. It's a metabolic disease.
Maria, we lost your screen. Can you share again?
So you see it, and you see it moving as well. Yes? Okay.
Yes, we do. Okay. Yes, everything's fine.
Excellent. Thank you. Good. The cachexia lipidome. And cachexia is a metabolic disease, and my group is interested in studying metabolic diseases. And it's associated with cancer. It's comes from the Greek words for a bad condition, and it affects 50 to 80% of all cancer patients, and is the cause of death for 20 to 40% of those patients. It's a huge burden to the population, and in particular to patients with cancer. However, cachexia also occurs in relation to other chronic diseases such as AIDS or COPD. It is characterized by a severe weight loss and wasting of different tissues of the whole body and a weakening of the body. And that's what causes the problem in the end. Its characteristic is an imbalance in energy homeostasis. And of course, you all know that energy balance is defined by energy input and energy output through nutrition or exercise or resting energy homeostasis. And in cachexia, this balance is unequal, and it's tipped toward the side of increased energy expenditure. And the problem is that we cannot simply let the people eat more to tip the balance again. This does not work for cachexia, and this really shows us that there is a fundamental reprogramming of the metabolism in the cachectic state that causes this energy imbalance. Cachexia is started by the tumor, of course, but then it affects many different organs in our body, including the cardiac and skeletal muscle, where cachexia leads to increased proteolysis and insulin resistance on the one hand, and reduced protein synthesis on the other hand. It also affects the liver, where cachexia is frequently associated with increased metabolic futile cycling, increased activation of the Cori cycle, increased acute phase response, and so on. And in the adipose tissue, it causes increased lipolysis in substrate cycling, increased thermogenesis and energy expenditure in the brown adipose tissue. It also affects the gut, leading to malabsorption, endotoxemia, altered microbiota, and bile acid composition. And importantly, it's also an inflammatory disease. So it really causes increased inflammation, systemic inflammation, also within the different organs. Cachexia is caused by either tumor or host-derived cachexia mediators, which we call cachexokines. And many of these cachexokines have already been identified, and these belong, for instance, to the TGF-beta superfamily, so myostatins or activins, but they also belong to the group of the pro-inflammatory mediators, such as IL-6 or TNF-alpha, which is also called cachectin, due to its very prominent effect on the different tissues. So all of these cachexia mediators have previously been shown to play an important role in cachexia and to influence the different organ systems to a different degree, thereby causing this fatal disease that is fatal because it reduces therapy efficiency and therapy tolerance of the underlying cancer treatment, and it also very strongly reduces the quality of life of affected patients and ultimately the survival time. So we are interested in identifying novel cachexia mediators, and we noticed that all of these proteins or all of these factors that are already known are actually proteins, but nothing is known about lipids so far, and that is a big surprise, because many different bioactive lipids have been already shown in a different context to regulate functions that play an important role for cachexia, such as regulating inflammation, metabolism, cancer growth, etc. So we set out to identify circulating lipids in relation to cancer cachexia and find a potential role of these important bioreactive molecules in this setting. So two students in the lab, Julia Zuber, who's a PhD student, and Pauline Morigny, who's a postdoc, they shared the work on this project, and they have looked at tumor implantation models, genetic models of cachexia, as well as patients with cachexia or who are weight stable, suffering from gastrointestinal cancer. They collected plasma samples of all of these different models and then analyzed them by LC-MS using the Lipidyzer platform. And this was done in close collaboration with Mark Hyde and Fabien Riol at the HMGU core facility. And this Lipidyzer platform is ideal to detect a huge range of different lipids, including neutral and polar lipids, triglycerides, free fatty acids, ceramides, etc. So overall, over 1,100 different lipid species in 13 lipid classes. Before I jump into the data, I thought I should perhaps briefly introduce you to the different mouse models of cachexia that we've used for the study. We found it particularly important to compare different mouse models and also different mouse strains, as we didn't want to identify specific factors that are only important for a specific mouse model, for a specific tumor type, or for a specific mouse strain, but that really had value in the more broader view. On the left side here, you can see that we have run 3 independent experiments using the so-called C26 model. In this model, colon 26 cells are subcutaneously implanted into bulb C mice, and then within 2 to 3 weeks, these mice develop weight loss or cachexia, as is indicated here by the severe weight loss, minus 12% roughly in the C26 cachexia group. As a control, we have a cell line that has the same origin, so it's also a C26 cell line, but this does not induce cachexia, and we called it NC26, and we thought this was a very important tumor control group to estimate the effect of the tumor independent of the weight loss of the cachexia. We had a look at 3 different independent experiments using this model, comparing either a very short time, so before the mice start developing weight loss, or comparing the NC26 and the C26 cells with cachexia, and then in a third round also comparing 2 different sets of mice injected with C26 cells, but this one group in the middle here was considered pre-cachectic because we analyzed it before it had lost any considerable amount of weight. This model relies on the bulb C mouse strain, whereas here we have another model where cachexia is induced by the subcutaneous implantation of Lewis lung cancer cells, so LLC cells, and these grow in black 6J mice. And then here we also have a genetic model of cachexia. So these were the different mouse models that we've used for this study. We have analyzed the lipid profiles in these different models, and I guess you can appreciate here on first sight in these PLSDA analyses that the lipid profiles in the cachectic mice were really clearly distinct from the lipid profiles of non-cachectic mice, either of the non-tumor bearing controls here in gray, or also of the tumor bearing controls without cachexia here in blue. And then this pre-cachectic group, before we could detect a considerable weight loss, they had a somehow intermediate profile here. We could also see that the lipid profiles in cachexia were clearly distinct by looking at these heat maps, for instance, here for the C26 model, where you can really see that almost all of the C26 mice clustered very nicely. We didn't see any major differences in the total overall lipid classes or in the fatty acid composition. However, we did see significant differences in several lipid species, and this is an example here for the C26 mouse model. However, I said in the beginning, we wanted to compare similarities between all the different mouse models. So we presented this here in form of a volcano plot, the 3 different cachexia mouse models. And I think you can appreciate that in all of the different mouse models, it became quite obvious that there was an increase in sphingolipid species, several different sphingolipid species actually, such as hexosyl ceramides, or sphingomyelins as well, that were increased in the circulation in all of the different mouse models analyzed. On the other hand, there was this decrease in LPC species that we also observed in all the different mouse models analyzed. And importantly, our negative controls were actual negative controls, so we didn't see this increase in sphingolipids here in the non-cachectic animals, as well as in the other tumor-bearing mice. We focused then next on the sphingolipids because we thought they were particularly interesting for the cachexia setting. They had initially been described as biomarkers for many different metabolic diseases, including cardiovascular disease or diabetes. And B, they also have very important functions that are critical for cachexia, such as being involved in regulating apoptosis, proliferation, stress response, or inflammation, just to name a few. So we thought focusing on sphingolipids was particularly interesting, also in the cachexia setting. What I should also show you here is that these specific lipids that we identified in the mice, we could also see in patients. So they are really hallmarks of cachexia, independent of the species we look at. And here we have a confirmation from the comparison of 20 cachectic patients with 19 weight-stable patients with the same type of gastrointestinal cancer. And here again, it's very clear that there is this increase in sphingolipids, as well as decrease in LPC species. So if we zoom into the different modified ceramides and look at the comparison between the different cachexia models, pre-cachexia, no cachexia, as well as patients, there were several that were significantly regulated in different mouse models, as well as significantly regulated in patients. And ultimately, we came up with a list of 6 ceramide and 2 LPC species that really defined cachexia, and those are summarized at the top right corner here. We selected them because they were significantly different in patients and also significantly different in more than one mouse model, as well as not changed or changed to a different direction in the non-cachectic tumor-bearing controls. The hexosyl ceramide 24-1 is one example that I wanted to show you some actual data. I use it as an example for all of these here because the same is true for all of them. They are significantly regulated in different mouse models of cachexia, as well as in patients in the same direction. They correlate very nicely with body weight loss in cachectic patients and also in the different mouse models we've analyzed. They are not significantly regulated in non-cachectic animals, and quite importantly, some of them were already high in a pre-cachectic state. And this made us think that they might have a good biomarker potential in mice perhaps, but also in patients. Pauline calculated the biomarker potential by calculating AUROC values of the different lipid species that we had detected. It was particularly clear for the modified ceramides that they actually have a very nice biomarker potential that is actually higher than the biomarkers that are currently used for the detection of cachexia, So what's the origin of the altered lipids in cancer cachexia? Are they from the tumor or are they from any of the other tissues? Initially, we thought that the adipose tissue might be a likely source. And the reason for that is the adipose tissue is one of the first organs that is lost, or one of the first tissues that is wasted, so that reduces in size during cachexia. You can see here that the loss in adipose tissue is really profound in the different mouse models. Here we had this key result where we found this drop in respiratory exchange ratio, indicating a switch to lipid usage in the cachectic animals. Also, when we looked at the adipose tissue of controlling cachectic mice, there was a really profound morphological difference in the adipose tissue. So the adipose tissue releases a lot of lipids, and we can also mimic this in the tissue culture by treating adipocytes with conditioned medium of tumor cells, which induce cachexia. We see that lipolysis, so lipid release, is increased in cachectic adipocytes on the one hand, and on the other hand, we also see an increased lipolysis, lipogenesis cycling, so degradation as well as buildup again. And this cycle then causes a drop in the ATP content, so in the energy content, which is what cachexia is all about, having a problem with energy homeostasis. We thought, yes, we can mimic this in the tissue culture, but can we also see this in mice? And indeed we could. We found this elevated circulating fatty acids also in cachectic mice, and we also found this drop in adipose tissue energy content in cachexia. And we hypothesized that this is due to an increase in the cycling of lipolysis and reesterification of fatty acids. And this not only then influences the cellular ATP levels, but may also influence the lipid profile, the circulating lipid profile of the animals and then also of the patients. Importantly, we have also found previously a new mechanism by which this cycling, this energy wasting cycling within the adipose tissue occurs. We found that AMPK, so the major cellular energy sensor, is very important here. We found a new way of preserving AMPK function in mice, and very importantly, and this is just a proof of concept study showing that if we improve adipose tissue energy homeostasis again. So if we improve AMPK function in the adipose tissue, we can not only preserve the tissue, so the adipose tissue mass overall, but we can also extend the survival of animals with cachexia. And this, and this was really particularly important and pointing towards an important role of the adipose tissue. And we thought that this might also be the place where the increased ceramides come from. So we had a look at ceramide synthesis and degradation pathways in the adipose tissue, and by looking at gene expression of the different key enzymes involved in all the different important pathways, and we found absolutely no change. Now we don't think that adipose tissue, despite all of our previous work on that, we don't think adipose tissue is the most important tissue here. We also looked at expression in tumor and muscle, and again here we didn't see any differences. However, we then found a significant difference in expression of many different key enzymes in the liver. Importantly, many enzymes involved in the de novo synthesis pathways are up on qPCR level, so on gene expression level, but also on protein expression levels, as you can appreciate here and here. We also see a change in expression of degradation and other important pathways, indicating that the whole metabolism and the cycling of ceramides is also increased in the liver. We have seen that we have increased ceramides in the circulation in cachexia. I haven't shown you the data today, but by now we also know that we also have increased ceramides or sphingolipid levels within the tissue, so in the liver, and this contributes to energy wasting, steatosis, and fibrosis in the liver, but potentially also acts on different distinct organs such as the muscle, where ceramides have previously been shown to influence AKT signaling, thereby mediating effects of insulin resistance or resistance to anabolic signaling, mediating protein synthesis and degradation rates, and they may also affect other organs such as the adipose tissue, regulating lipolysis or fibrosis there, and they have also a prominent inflammatory role. We've now started the process of finding out what exactly the role of the ceramides is in cachexia, because it's a really robust finding that has now also been confirmed by other labs that the ceramides are high in cachexia. And with this, I'm at the end, and I would like to thank everybody involved in these studies, in particular Mauricio Beriel-Diaz, whose group has run this study together with my own group, and Mark Hyde, Fabien Riol, and Jerzy Adamski from the HMGU core facility, who have been extremely helpful in performing these studies. And yes, thanks of course to my funding agencies, and thank you all for your attention, and I'm happy to take questions.